wait a couple minutes for
people
to show up. Yeah.
Sure. Oh boy. Back to paper writing.
Yeah. So I'm waiting for one of our our presenter today.
Oh, right.
He's going to come in. There he is. Ujjwal. All right. And I have, so you're ready to, I want to go over a couple things before you present. So, but can you share, we can test your screen share when we get to that part of the meeting, so.
Yeah, just one.
Okay. All right, why don't we start the meeting? Welcome to the meeting this week. I know everyone's kind of jumbled around because of the coronavirus. I'm out of my meeting space right now, my usual meeting space currently, but it seems to be pretty okay for right now. So why don't we go over a couple things before we get to Ujjwal. Ujjwal is going to present his proposal for, he's going to give us an update on what he's been doing recently in terms of 3D modeling. He also has some proposal for the one of the GSOC projects, the one where it's a lot of revamping of the DevoZoo site. So we'll go over the message he sent me in Slack, but there are images involved. I wanted to get feedback on that. So let's see. First of all, does anyone have any comments or questions or news?
No, it's amazing you're doing this.
Yeah, it's all the rage now, apparently, meeting virtually, even though I've been doing it for a couple years. So,
yeah.
So why don't we go? I wanted to cover a couple of things before we move to Ujjwal. The first one I think I'll talk about here is I found an interesting paper. Let me share my screen. I found an interesting paper on C. elegans. The group is called DevoWorm, but we've been interested in other organisms as well. And so we've done C. elegans, we've done some work with Drosophila, and there's some data with zebra fish on our DevoZoo site. And we've also worked on something called Tunicates or sea squirts. And it's a pretty big group of organisms, pretty diverse, but it has a really interesting developmental period. And so we've done a little bit of data analysis on that. And I found a paper that it's a very longish paper, and I can share it with you. It's in this folder, and it's on the eventful history of the non-embryonic development in tunicates. So tunicates have a very interesting embryo, and it's actually a lot like C. elegans in that you can take the individual cells and trace them from a single cell into an embryo of a couple hundred cells. And so you can they have the lineage tree traced out. And from there, you can do a lot of things in terms of analysis of the embryonic development. It's also interesting because it has a very small connectome. So in C. elegans, we talk about the connectome being 302 neurons. In sea squirts and tunicates, they have 177 neuron nervous system, at least in the developmental phase. And so that's another thing that I don't think anyone's really quantified it the way they've done with C. elegans. They know the number of neurons, but they don't have them cataloged. If you know anything about OpenWorm, the more general OpenWorm project, you know that there have been a couple of draft connectomes where people have actually really characterized each cell in the connectome. They don't have that for tunicates, but they do know that it's a small connectome. So that's another advantage to working with tunicates. The third one is that they have this extensive non-embryonic development phase. And this phase ranges, it grows from maybe a couple hundred cells to over, thousands of cells. And it changes shape significantly during this non-embryonic development phase. And so this is what this paper is about. And they kind of review a lot of this part of the life history of this. It's a pretty diverse group of organisms, but they just kind of do a general review of it. So tunicates encompass a large group of marine filter-feeding animals. More than half of them are able to reproduce asexually by a particular form of non-embryonic development, generally called budding. And so the phylogeny of tunicates, which means their evolutionary tree, suggests that asexual reproduction is an evolutionarily plastic trait, a view that is further reinforced by the fact that budding mechanisms differ from one species to another. And so there's a lot of diversity in that budding mechanism. So even that within this order is quite diverse. And so that means it happens different ways in different species. And so they review 150 years of literature in this review to provide a view of non-embryonic development, which they abbreviate NED, diversity. And we present a comparative picture of budding tissues across tunicates. So this is mainly having to do with that budding period of this non-embryonic phase. And they're just kind of reviewing a lot of the evidence for how diverse it is and the basic mechanisms and so forth. So this is, they get into this sort of introducing us to tunicates in this part of the paper. They typically have a biphasic life cycle with a planktonic larva in a post-metamorphic phase that is benthic in the Cydians. This is probably to a non-specialist, this doesn't make sense, but it basically says that there's this life cycle where they have different stages. They have a larva stage and a post-metamorphic stage. So it's sort of like in butterflies, they come from a larva stage and then they transform. That's kind of what happens in tunicates, although not in the same way. While solitary tunicates reproduce strictly sexually, many can propagate both sexually and asexually. So that's actually like C. elegans, where we have sexual reproduction in some cases, which happens because there are males in the C. elegans population. It's at a very low frequency, and they can produce sexually, which usually happens when there's in times of stress or times of drought where the worms are stressed out and trying to survive. They can sometimes reproduce sexually, but they reproduce sexually in general at a very low frequency. But most of the time they reproduce asexually, and this is exactly what happens in tunicates as well. So in budding, this process that they're reviewing, they bypass fertilization, embryonic development, larval stage, and metamorphosis. And in the budding, when these buds bud off and they break away from the main organism, they give rise to one or several adult individuals called blastoids. And so there are these little buds that come off. They're like, you can think of them as clones, and they disperse and they form an entire organism. If you think that's weird, I guess it is, but it maybe not. because tunicates are a very large group of organisms. But that's the way they do some of their, they do reproduction that way as well as sexually. Let me let Susan in. Susan's here.
Oh, good. Hello, Susan.
Hi, Susan. So, Susan, I'm talking about non-embryonic development of tunicates right now. I'm kind of going over this paper. So it's I'll share the paper with everyone, but I'm kind of walking through it. You might find this interesting. So they go over this, and I don't want to get too deeply into the paper because you can read it on your own. And so budding and non-budding species are scattered amongst forming tunicate orders. And so an order is a group of species that are related loosely. And there are 4 orders where you get both of these types of reproduction. And it's not like typical of like one group of organisms with a common ancestry or maybe one order. It's scattered throughout. So that the implication there is that it's not something that's occurred in one ancestral species and then descended into its descendant species. It's something that happens. It's what they call polyphyletically, or it happens just kind of based on the species, or they may be based on a couple of species and their conditions. So it could be something that's influenced by environment. It could be something that just happens as a strategy that they have that's dormant in other species in that order. It's definitely not a what you would call a recent adaptation. So last few decades, the study of tunicate budding has focused on a few model species. So they have model species, like everyone uses a model species. It's usually a species that we know that people work with in the lab, say, or that's common enough in nature that people can get a bunch of them to study in a lab setting out in the field. And but that, of course, limits our ability to understand the diversity of these processes, because a model species, like say C. elegans, represents a little bit of biology. And we think of C. elegans as like the nematode, right? Now there are a lot of nematode species. They range very broadly in terms of the number of cells they have, in terms of the types of reproduction. Some are sexually reproducing exclusively, some are exclusively, hermaphrodites, etc. There's a lot of diversity there. But we usually just use the C. elegans model, and we use it to say things about human biology. So, it's not just that we use it to look at nematodes, but so model organisms are, definitely they have their place in biology, but it's important to recognize that they're a very limited view of life and its diversity. So they actually go through tunicate phylogeny, and this part may be a little over the head of a lot of people in the group here, but phylogenetics is kind of one of those things where they throw around a lot of jargon, and you have to read a couple papers on it to know what they're doing. But basically, what they do in phylogenetics is they study. So this is a tree of a bunch of tunicate species, and you can see how it looks like a binary tree. If you're a computer scientist or if you're a biologist, you know that this is a phylogenetic tree. It's just how species branch off from a common ancestor. So these are two species, and this is the common ancestor. And the common ancestor doesn't actually have to exist in nature. It's just inferred from what you see in the species that are living. So a bunch of these species were observed, their traits are known, and then you reconstruct these common ancestors from how similar the species here at the tips of the tree are. So these species are very closely related. this species is related to these two, but not as closely, and so forth. And you build these trees by analyzing traits, and you get this tree. And the tree then can be, parsed in terms of its, different groups. They call them clades. So this is a clade, and it just means that it's a single group that you can find like two sister species, and maybe like a third species that's here, and another species that's out here. There's, definitely some subjectivity in how you define a clade, but basically there are these groups of nested tree branches. And so this is a tool that they use to understand how to group organisms by order or by genus. And, those organizational schemes came before phylogenetics really became a thing, but that's the way they kind of understand a lot of diversity between species in biology. And so there's a whole field dedicated to this, and they really get down to the nitty-gritty in terms of analyzing these trees. But they talk about it a bit in this paper. So definitely, if you don't understand this part of the paper, that's fine. And then we have some pictures here of some tunicate development and some budding strategies. So we have a couple of nice color photographs of different tunicates. So as you can see there, if you know anything about tunicates, they don't look like this in their adult phase, but they look like this in their what you might call their larval phase. So they have the embryo, which is this little ball of cells, and then they form these structures here, and then they eventually form something that actually is free moving in the ocean. And so they don't really resemble their adult form here too much, but they don't really resemble their embryonic form either. And so you can see in this case where they show the budding process, where they have these larval stage things and they're budding off new buds, and then those buds will become new larvae as well. Let's see. Here's another image. This is another set of images that show some more examples of larvae. Again, you have this tube. So this tube will eventually become a free-moving organism in the ocean, but it's just a tube in the larval phase. And then you have these different strategies for budding again. And they show it at the cellular level, where you have buds that are forming, you have vesicles, you have a tip, a primordial tip, and this kind of shows this process. And then they kind of go over what we don't know about this process. So there are a lot of orders. Like I said before, the model organism is a good way to characterize the process, but not a very good way to characterize the diversity. And so we have there are a lot of orders and groups of this phylogeny for which budding is very poorly known. And so there is a lot of diversity in this budding. I think these two figures 2 and 3 show the differences, what they're trying to get at. So this is one type of budding here, and then figure 2 is another type of budding, which is a little bit different, prairie branchial budding versus stolonial budding. And so it's a very different process between the two. But then, of course, across orders and across groups of species, it's not very well known how this process happens. And so our knowledge about certain types of budding come from certain model organisms, and so we don't know the full details of how it works in nature, just kind of in our model organisms. What else? Let's see. And this is budding in a specific type. It's a colony of a certain group of this organism, and it has this kind of shows another couple color plates and more of this process. So they show how it happens in a certain group. What else? So they go through these different types of budding from bloom to bud, and then they get out. There's some anatomy in here, and so then they get into reconstructing phylogeny. So they actually talk about something called homology. So homology is when you have two species that are related and they share a trait. So the common ancestor had this trait, and then it exists in the related species that were derived from this common ancestor. And so you can call something homologous if it's shared within between the two sister species. Now, homology is interesting because you can have mutations that occur along the way. So you can have some process or trait that looks similar, like bird wings across birds, but they function differently. They may look different, but they have basically the same homologous relationship. And so this is one thing they use to understand common ancestry and infer common ancestry. And so, like amongst phylogeneticists, homology is a big issue. Is something homologous? And it's like, so you would have like a homolog would be like a certain type of budding, but it's maybe a little bit different than its sister species, but they're both common. They both have a common origin, and so they talk about this non-embryonic development and how homologous it is across species. And so then they have a table. There are actually 6 budding modes in Tunicates. They talked about the first 2 here, or the stolonial and the peribranchial, which are the main ones, and then there's vasovascular, epicardial, and pharyngeal. And so all those different types of budding were identified in the literature, or have been identified in the literature, and they give example species of each. So this is, and then of course Tunicata, which is this big group of Tunicates. This is something that, if you're interested in the evolution of development, is a great model system for this. And so this is, if you're interested in the theory of it, or if you're interested in this topic of Evo-Devo, this is a section you should read over. They're proposing that it be used as a model system for Evo-Devo. And certainly, the evolution of development is an interesting area we don't talk too much about in the group, but it's where you look at development and you try to figure out how basically development evolves, or how development can contribute to evolutionary change. And so there's a huge literature on Evo-Devo, and there are a lot of model systems that exist now, but there's definitely some room for new model systems in that. And so these are the references, or a lot of references, because it's a huge review. And that's the paper. So let me share it. I'll share the link. And so that's in that drive, and if you need access, just sort of request access. I think if you can't access, it'll ask if you want to request access. The next thing I just wanted to talk about briefly before we move on to Ujjwal's presentation is that, and this is for Dick's interest here, we talked about, actually, we exchanged some emails about the embryo physics course. And so this was something that happened actually quite a while ago, anywhere from 5 to 10 years ago. And it was something that Dick hosted on Second Life, and the virtual reality platform. And people would come in and they would give presentations on different topics. And that stuff is now on the web, hosted by one of the participants from back then. He still has it online. And what I've been doing is I've been going over and trying to archive it as an educational resource. And so what I've been doing is just kind of pulling it over from that site and putting it into a GitHub repository. Now, and this is just kind of early days on this. I've not really done a lot of work on this, but I'm trying to thank you, great job. I'm trying to pull it over into this repository. So the first step is to get the presentations down and into this place. And then this is a lecture tab. So this actually just has a list of the lectures with the abstracts. And so this is kind of like right now, it's a mirror of the other site. But what maybe we can do with this is like turn it into a resource for education. And so we have the slides, we have all the materials here. So what we have are like, different years. I think it goes back to 2009. Yeah, it goes from 2009 up to 2014. And each year there were a series of lectures by people in developmental biology and biophysics. And they have a talk, they'll have an abstract with slides. And, there are a lot of things we can do with this. We don't just have to have this list of slides and videos. We can build maybe tutorials around it, or kind of profile different topics or things like that. So right now we have this little stub, and we have the presentations. But I'm thinking kind of like back to the DevoWorm OpenWorm education thing that we talked about at the beginning of this year. That was done last year for Mozilla Open Leaders. It's something like that. We can talk about it later, but where we have like the presentations, the lectures broken out maybe by year, and then maybe some tutorials to go along with it. Kind of turn it into sort of a educational experience rather than like a list of lectures. I'm not sure how that would best be approached. I mean, that would be something that would have to be discussed amongst people, what they think would be interesting. I know that we have a couple people here who were involved in Open Leaders when I was, Yash and Devansh, and so they'll be interested in maybe giving some feedback on this. Jesse is also interested in this type of educational, online educational thing, so he might have some interesting comments to make on that. Why don't I put a link in the chat, and you can look it over. Yeah, so Dick says the other site is temporary, and that's why we wanted to originally mirror it over to this one. But we can also build up some infrastructure around this set of lectures, because we already have a lot of the content here. We have presentations and a lecture series type thing. There are topics, and there's a lot of information in here. For the casual viewer to come in and to read this, it might be a bit overwhelming, but that it might also be very informative. And you have this sort of fire hose situation where you're trying to get all this information down from the web, So to make it a little bit more structured too, I think is the key. And so now, Ujjwal, we're ready for your presentation.
Yes, sir.
Okay. Why don't you share your screen? Oh, wait, I gotta stop presenting now. Okay. Go ahead.
Am I visible? My screen is visible?
You can see your screen, yes.
Yeah, so today, I'm going to talk about CompuCell3D computations. So basically, CompuCell3D software is a kind of software which is used to simulate, build, test, run, and visualize multi-scale, multi-cell GGH-based simulations. And so, we can start with the introduction. So, CompuCell3D, as I've suggested, a three-dimensional C++ software program solving environment for simulations for biocomplexity problems, integrating multiple mathematical models. These include cellular pods models, which can model cell clustering, growth, division, death, adhesion, and volume and the surface area constraints, as well as the partial differential equation solvers for modeling reaction diffusion of the chemical fields and cell type automata for differentiation. Now, it has the built-in standard scripting language Python that allow the user to quite easily write extension modules that are essential for building sophisticated biological model. As we can see, like it is not a hard build model, so it does not require specific biological systems to run. You just have to code in the certain languages. Also, it does not require recompilation again and again. Like once you have write the code, you can make changes in the code and can run it simultaneously without recompiling again and again. Model I use these. The best part of this CompuCell3D is like these models described using XML and Python scripts. These are something which we are focusing since the last summers. Like we are trying to extract XMLs for the basal area model and try to find out the positions which can be easily fed in this type of simulation softwares. And it has its specialized editor, TW edit, like we are going to talk about it later. It has multi-platform support like Linux, Unix, Windows, and Mac OS. So for here, some about like installation guide. So in the for the Windows, the installation is pretty much very easy. Like I recommend everyone to please, if you have Windows, try to do it on Windows, because in Linux it is very much very much difficult to install it. So what you have to just go to a CompuCell3D's website main page. There is a menu in the left side page of it. Click on the binaries option. You can see the different download options for different platforms like Mac OS, Linux, and Windows. For Windows, click on the download button. It will once you find download button, it will redirect you to the third party site, which will help you to download it because of their good servers. And it is around for 70 MB or something. And you just have to, once you have installed just have to execute the exe files. Like if I want to show you some, like this is the CompuCell3D XML files, and you just have to click on it. So these are the simple steps. So you have to just specify the location and you have to click on install. Like I have already installed it. So I'm going to do it again. Linux installation is very tedious. Like there is, I have also like mailed the submersion. There is not proper documentation for Linux installation, and there are many libraries which are outdated, due to which these are causing problems in installation. By the way, the process is same. Like you can find the Linux source from the download link, and then you have to unzip it and follow the PDF. So I have downloaded it on Linux as well, and I have found out the problems which are there. So just in case you are using Linux for this, I would like to make sure that you are not going through the problems. So in this command, there is a CMake GUI which is downloaded using Swig. So this is the old outdated version of CMake GUI which is not going to work today. So what you have to do is, once you have installed all these files, you have to install this one, Python Qt graph for simulations. But at this step, at CMake GUI, you are very much comfortable. You can set where is the source code, binary files, all these options. But once you create configure, it will show you some errors like CMake 3.13 or higher is required, and currently it is installing CMake 3.10. So you have to install the whole CMake again from the CMake library or CMake website. It will give you some 3.17 version. Then you are again going to encounter a problem because CMake GUI is outdated now. CMake GUI is itself a model for making this box. So you have to update the CMake GUI as well. Once you have done that as well, you are again going to encounter a problem, that is the outdated version of VTK library. So VTK library and a Python version may not match, which is going to cause some sort of problem. So you have to go to the VTK library files and you have to edit. Like I have compiled a document for installation with all these corrections. I will share it with you on Slack platform or something. So this is about Linux installation and problems with it. So building our Fediverse simulations, first of all, we have to familiarize ourselves with the environment. So this is how the main screen looks. Like it has some options like file, view, simulation, visualization. It is more like any other general editor I'm going to. So how you run to a first simulations, they have some demos which are already installed or while you are installing CompuCell3D. So you can check out these models if you want to run. Like I have loaded one of the tumor models. So it is how these precisions are being seen. EWEdit++ is a custom editor for the CompuCell3D. Basically, CompuCell3D is not something like you can build models by drag drop. It provides you some essential, some prerequisite features of the model which you are going to create, and then you have to write the code like simple C++ code to make things happen. It is easy because it supports Python with C++, and as I have written, like you can use this editor to write your own codes for the running your simulations. You can define your hyperparameters, shape, size, movement, etc. for the simulation using this editor. So there is a quick demo like how to run the software initially. So you are in the CD plus CD3. This is the text editor. I have created an example for today's session. Like this is something which I have created. So let me show you before explaining how the process is being run. Sorry for the interruption. I don't know. Let me start it again once if it is not responding. So you have to go to the command prompt. You have to go to the command prompt. You have to copy the location where this is stored. For example, in CompuCell3D, you have entered in the CompuCell3D folder. After that, you have to run this file, CompuCell3D bat. Once you have run this file, it will open another. It will open another window. So this is your CompuCell3D player. So like if you have opened it, we can give it. So you can open the simulation files which you have created here. For example, I have created an example file for this. So if I open this simulation file, you have to press run. Once you have pressed run, it will create a simulation. So this is a basic simulation for condensing and non-condensing cells. I have provided the various parameters for this. So there is a now. If we stop with G to stop this simulation running, you just have to press cross button. That's it. Stop, pause, stop. Now we are going to the TWEdit.bat. So this is the inbuilt editor as I have shown you. So you can go to file, open. You can click on example to open. So this is one of the file. So in this way, like you can open the folders and it will open the like expenditure file. So this is the file, and I'm like trying to show you uh, XML document mainly. Yeah. So there I have specified the different medium of condensancy, non-condensancy, which you can separate out here. So this is like this file is basically generated as you have specified the parameters in CompuCell3D environment when you are creating a new project. Let us try to create a new project itself. So if you go,
If you're tired, you should stop and go like that. Who are you talking to at 10?
Very well.
Okay, just,
If I get through them,
so this is how you are going to for simulation. So like why we are approaching CompuCell3D is the main purpose. Like we have extracted the XML documents and all these things. Also, the basically also 99% of the modeling done with the custom little code. Like we are easy to share code across the multiple platforms and use these standard modeling tools improves the chances that research being accepted and further refined by the other scientists. And these standards generally improve the research efficiency, and like bug tracking and all these things are easy. Like we are trying to build simulations for basically Bacillary and C. elegans. So I'm in the exploration stage of the software. I am working with the software for about a week and I'm still exploring it. I'm not very much familiar with it right now, but as the time passes, I will get to it. Also, also in the future steps in the coming meeting, like on by the mid-April, I'm hoping that I will run this software completely and I will try to develop some basic primitive types of simulations. Like today, which I have shown is a very primitive type. It is not something which we are going to be satisfied with. We have to build specific complex problems. And as the GSA project 14 is concerned with the PTN model project, so as we get the more and more data about the movement and all these things, I think we can very much use this software to create the simulations by the end of GSA period. So that's all on my side about.
Okay. So I have a couple of questions here. One I think was Dick. He said something about can this program handle differentiation waves? I know that like when I actually visited the group a couple years ago, and they said that they showed me some of the models like what it can do, and it does handle a lot of dynamics. So what you do is you build these cells, and you can model the cells, form different tissues or whatever you want to form, and then you can animate the cells themselves. So you specify the cells with information about like what their content is, different receptors, and then you can model things that go on in the cell, all sorts of processes. I think they were modeling things like the liver, and they were modeling embryo. There are some embryo models that people have published. So I think they can probably handle it. It would be, it'd have to like think about how we might want to represent it, but I think it definitely would handle that. There are a lot of there are a number of publications. If you go to the website for CompuCell3D, they have a bunch of publications listed, and so they've done a range of different types of studies with this platform. So Susan asks, does your program include forces like pressure and strain? Yes, it's physical. There are a lot of physics that it models. So they're largely interested in modeling all sorts of physics going on in different tissues and organs.
Basically, to answer this, Susan ma'am, so basically it is a raw software. Like you can include every aspect of what you want to do, pressure, or all these things using C++ code. All you have to do please define some constraints. You can include all the simulation that you can do. And this is the best part of this software because it is not something which you have to do, which is predefined, like you can do this. You have the software, you have parameters, you can define parameters. Like if I can show this, so if you run CompuCell BAT, and when you are creating this new project, for example, if we start a new project, basically I am in example 3D and I have to so basically, like if you are trying to create a new software, I am not currently able to get out of this example problem. But if you are creating a new problem, it gives you certain set of rules, like what you have to. Oh, just a minute. Yeah, so it is in the toolbar. Sorry, when you go and create a new module, you have to write the module name. You can certify certain layouts, certain plugins, certain energy functions, and once you have created this, it asks you for the different models, like for example, we create for demo. So it asks you for the lattice dimensions, like what should be the graph dimensions for this. It asks you for the boundary conditions, like what are the average membrane fluctuations, pixel copy range. It gives you like a lot of prerequisites which are going to be needed for your purpose. You can add specific cell types, like if I type condensing, so you can add these specific cell types, and after that you can specify in the code like what you want this cell to be doing. Freeze option is to basically freeze this cell, and so that it is not going to be fluctuating in the other simulation that you are going to create. After that, you can chemical field names for the different. I am still figuring out like what we can use
Yeah.
with the chemical fields. And you can also adhesion forces like focal point plasticity. It has explanation like what you're going to include in your thing. I have for the project I have like included a context, volume flex, and next, and you have to click on finish. And here you can see all your files like which are already made. You can add as many as parameters that you have in this section, and you can create. So yes, the answer to your question is yes, we can include all sort of uh, constraints like pressure and forces and in the software as we have to just code it out.
Yeah, I think that's very good. So yeah, this is based on something called the cellular POTS model, which is like it's like a cellular automata, but it's not quite like that. But it's it's something from physics, and they use it to sort of, you have these cells that are arrayed in different locations, if you're building like a tissue or an organ, and then you specify the relationships between the cells, and they have all these built-in functions. So that's what makes it attractive, as opposed to using something like Morphozoic, which we saw in a past meeting where we have a cellular automata. We can build like something that is able to generate patterns and other things like that, but it can't implement physics routines that we might want to do if we want to model things like biological processes. So each that's a good lesson that each type of model as implemented will have different strengths and weaknesses. not just, you can't just throw any modeling platform you want at a problem. I think, like I said, I visited the group that made this software a couple years ago, and they showed me a lot about like what it can do, but I've never really had the chance to develop it very much. I think it would be a good opportunity for like someone like Ujjwal. If other people want to participate, they're welcome to. But to have this platform where you can build models of embryos and things like that. So Susan said I would like to try CompuCell3D. Yeah, it's downloadable from the web. I think it's cc3d.org. Yeah, Jesse. And they have, so Ujjwal, can provide slides, and he can provide maybe I don't know if he has a link. I can provide a link in an email after the meeting. But it's so you can download the package, and it's, it may take a little bit of effort to get it to run. But like I said, they have on the website, they have a bunch of, they've used this in publications. So for different things like embryos and models of organs and other types of biological problems. And so if you go to those papers, you can often find out exactly how to implement it for a certain type of thing. So that actually might be a place to go to if you're not really like a software guru. So try the Windows version, I guess, as is Ujjwal's recommendation. So yeah, that's good. Well, thank you, Ujjwal. And I'll send out an email after the meeting about this to give you a little bit more background on it. Finally, Ujjwal, I had your you sent me a message on Slack about your proposal for GSOC, and you included some images. I just wanted to comment on those a little bit. Do you have that up? Because I had it up, but I lost my browser window.
Yes, sir. Just a second,
Okay.
Yeah, so these are the images that I'm talking about.
All right. So this is for the project where you're sort of refactoring DevoZoo. That's what your proposal is, or refactoring means like revising it. So the first picture, actually, go back to the first picture. I was going to make some comments. So this is interesting. It's good, except that I was wondering if we could replace the full worm with like an embryo of some type, because it's development oriented. I mean, open worm projects, probably be DevoWorm projects. Although I think we'd also have a link to open worm too.
Okay, sir. I have noted this comment.
Maybe I'll send you some mock-ups like on Slack. I'll draw them up and send them. But I like the idea of having like an animation in the background. But I wanted to figure out how to make a back animation that's that would be the project, right? Coming up with good visuals. So
I'm also thinking we can use the time lapse video that we have on YouTube for C.elegance as a background video, by compressing it further, so it will make a good animated video for.
Oh, that'd be good. Yeah.
/page.
Yeah. But yeah, I mean, also, yeah. So the number 2 is this. So this is I guess an array of icons. I like it. It's a little busy, but I mean, they probably can be organized a little bit better. But I like the basic idea. Number 3, this is the mission. Yeah, this is good. Just kind of going through microscopic tutorials. So this would be like, tweets. Can you go back to 4? Yeah, so this would be the tutorial, like microscopy tutorials. So this would be like, Yeah, there is a page where we do that. I would like to have, yeah, like a page of tutorials, maybe with some icons for the links. Yeah, well, we can talk about that. And then number 5. So yeah, we don't have like a Twitter account for DevoWorm per se, but we can pull in OpenWorm. Usually that's handled though, like in a little, like a window, like a plugin app, like on the front page. Like on the OpenWorm site, there's like something like this. This might be a bit better to handle it, but okay, Ujjwal, yeah, that's good. Why don't I mock up some of those in drawings, and I'll get back with you. And if anyone has any comments, you can have them. You can make them. So let me go through these chat messages before we go. So we have a bunch of chat messages here. We have people say they want to try CompuCell3D. Dick is getting tired out. Well, okay. We're at the top of the hour, so. Linux installation, advice and updates on pandemic. So this is a link Dick shared with the COVID-19 information. Please check it out. And this is, images look good, Yash says, so thank you. Okay, well, it's at the top of the hour. Any other questions or comments before we go? Okay, I just wanted to alert you that next week I'm going to cancel the meeting. I have another engagement. If you want to use this link to meet on your own, you're welcome to do it or whatever. But we won't have a formal meeting next week. And then we'll pick up the following week. I don't know what we'll do yet. I can get some, feedback. Maybe someone wants to present something. Yeah, so Dick says, keep in touch, RE CompuCell3D. I think we'll revisit this maybe in April sometime, when maybe Ujjwal will do a little bit more work. We have some people interested in looking it over and seeing what they can do with it, so we'll definitely revisit it. Okay, thanks for meeting. Glad you're feeling better, Dick. Okay.
I've been tired.
Yeah, well. Okay. Oh, April's exams for Susan. Okay, well, it'll be, we'll work on it. It's, we're not stuck to a schedule here. Okay. Well, good luck on the exam, Susan. Okay. All right, thanks. Talk to you guys later. Stay safe.